Okay, so we've given people some time to join into the presentation, so we'll go ahead and get started. Hi, everyone, and thank you for joining today's webinar brought to you by Microfluidics in collaboration with Sky Pharma and LAGEP, where we will be discussing an innovative solvent-free technology for oral bioavailability enhancement. My name is Chris Jaquin, and I will be serving as today's host as well as a presenter, and I'm an applications engineer at Microfluidics. And to start, I'll briefly give some background about who Microfluidics is as a company. So we were founded in 1982, and we have been manufacturing high-shear fluid processors utilizing our fixed geometry interaction chamber technology ever since. We are headquartered in Westwood, Massachusetts, just outside of Boston, with localized support in 47 countries, and to date we have sold over 4,000 processors to over 2,000 companies. Around 10 or so years ago, we were acquired by the IDEX Corporation, and we have since been grouped with our sister companies, Quadro Engineering, Fitzpatrick, Cerados, Matcon, to form the Materials Processing Technologies, or MPT platform. Our processors are used at a variety of scales for a variety of products, including being used for both R&D and manufacturing of APIs, vaccines, inkjet inks, coatings, nutraceuticals, cosmetics, and much more. Over the past 40 years, we've amassed a vast knowledge of both applications and machine design, and we like to use this knowledge to aid our customers. So we really like to point out that our customer success is our success, and we really aim to work with our customers to ensure that they are getting the most out of their microfluidizer processors. So with that being said, I will let the other presenters introduce themselves and their companies.
Hi, everybody. My name is Vanessa Bourgeot. I'm Innovation Leader at Sky Pharma. Sky Pharma is a French CDMO founded in 1996 and independent since 2021. This is a center of excellence in oral solid dosage forms. We have roughly 150 employees, including 1/4 in development, and also we have 5 PhDs. And we have developed over the years an international business and partnerships. So our manufacturing facility and development is based in Lyon, in France, only 20-minute drive from the airport. And we are able to cover a full range of services to support pharmaceutical projects. It can go from formulation development, lab scale prototypes, and QBD studies, and also scale up manufacturing of GMP clinical batches and stability studies, and after a routine industrial manufacturing as well as packaging. So I will now hand over to Oksana.
Hello, everyone. I'm Oksana Lemasson. I'm PhD student at LAGEP, which is a multidisciplinary laboratory based in Lyon. Our main research activities are around 2 scientific topics. First, physical-chemical processes in complex, dispersed, and involving media. And secondly, dynamic modeling, observation, and control of processes. We are actually developing numerous academic and industrial collaborations. And each year, more than 80 publications are done, and 12 to 15 PhDs are graduated also.
Awesome. Thank you, Oksana. And before we dive into the bulk of today's webinar, we will briefly touch on today's agenda. We will begin by discussing some bioavailability concerns with specific types of APIs, before I will give a brief introduction to Microfluidizer Technology, and then handing it over to Vanessa, who will briefly introduce both IDD Dissocube and NanoMics technology. At this point, Vanessa will give a brief case study regarding IDD Dissocubes and Fenofibrate, before Oksana will discuss a separate case study around the NanoMics technology with 2 different APIs. At that point, I will briefly discuss the microfluidizer processor scalability, touch on our product line, before we get to today's take-home messages, and then again, at the end of the session, we will leave around 10 to 15 minutes for a brief Q&A session. So if any questions come up throughout the course of the presentation, again, please feel free to ask them throughout the chat feature throughout the presentation. So with that being said, I'll turn it back over to Vanessa.
Thank you, Chris. So let's start with a very simple definition. Bioavailability is actually the fraction of an administered dose that reaches the systemic compartment. And for an oral dose, it is calculated as a ratio of the area under curve that is obtained after oral administration over the area under curve that is obtained after intravenous administration. So we can easily understand what it is so important to reach a good bioavailability for an oral drug. However, ensuring sufficient and predictable systemic drug exposure is not straightforward, and bioavailability issue is one of the top reasons for attrition of oral new drug candidates. Next slide, Chris, please. So, what happens to a drug that is orally administered? First, the drug may exhibit solubility issues that may be related to its physical properties. For instance, log-P, pKa, particle size. Along its journey, the drug may also be degraded through pH variations or surrounding enzyme. But the main bioavailability impacting factors are known to be related to PGP, P-glycoprotein, as well as first pass effect. So PGP is a transporter that pumps back drugs into the lumen, therefore decreasing their absorption. And cytochromes are involved in the first pass effect, are actually responsible for drug metabolization in the gut wall, but maybe in the liver. And on the top of that, there is a wide variability of this expression of those transporter and enzyme that may lead to a huge variability in the clinical response. That is why for almost all of the oral drugs, very low fraction access to the systemic compartment where it must have its activity actually. Next slide, please, Chris. So for those who are familiar with this classification, there is actually a biopharmaceutical classification system used to range categories of small molecules according to their intestinal permeability as well as their solubility. And the class 2 and 4 molecules are the one which exhibit the most poor solubility, and it is estimated according to the industry that almost 80% of drug candidates are poorly soluble, so need to be reformulated and potentially using microfluidizer technology. And Chris will explain further how this technology may be helpful to enhance the bioavailability of a drug.
Yep, thank you, Vanessa, for introducing the potential need for microfluidizer processor technology. So to start, I'm just going to briefly walk through how exactly our technology works by going through this brief schematic. So to begin, you would premix your formulation before introducing it into the inlet reservoir. At this point, it would be pumped via a constant pressure intensifier pump through a fixed geometry interaction chamber at pressures of up to 30,000 psi before passing through a temperature control unit such as a heat exchanger. The core of our technology is this fixed geometry interaction chamber. Housed within this interaction chamber is a small microchannel, and essentially how the technology works is by forcing your material through this very small orifice at very high pressures, we are able to create very high shear forces along with very high impact forces as well. So we offer two different types of interaction chambers, two different geometries. We offer a Y-type interaction chamber, which we typically would recommend for any liquid-liquid systems. So think liposomes, emulsions, lipid nanoparticles. And then we also offer a Z-type interaction chamber, which we typically would recommend for any products that contain solids. So think suspensions, dispersions, cell rupture, applications of that nature. So how the two different interaction chambers types work. For the Y-type interaction chamber, your product would enter via the high pressure inlet before separating into two separate streams. These two streams would then both pass through separate high shear zones. So this is the actual microchannel itself. Before the streams then impact upon one another, impinging in this high impact zone before then exiting the low pressure outlet. So again, there's really two major forces at play, the high shear forces and the high impact forces, which are utilized to reduce either your droplet or particle size. The Z-type interaction chamber works slightly differently. So rather than having your product split into two separate streams, the product is maintained in one stream. The entire stream will then pass through the high shear zone, which again is the microchannel, And then in the case of the Z-type interaction chamber, rather than having these two streams impinge upon one another, causing these impact forces, the impact forces are actually up against this back wall. So again, there are still shear forces and impact forces at play. It just works slightly differently for the Z-type interaction chamber. So I mentioned the two different types of geometries. We offer a Y-type interaction chamber and a Z-type interaction chamber. But it's also important to note that these interaction chambers come in different dimensions as well. So I've listed here on this slide some of the more common interaction chambers that we offer. And really the point of this slide is to show you that by selecting a specific interaction chamber and processing your material at a specific processing pressure, you can see that we are capable of reaching a wide range of shear rates. So really, regardless of what your product might be, we should be able to apply the necessary shear rates needed in order for you to achieve the desired results with your product. So microfluidizer processor technology kind of works on the basis of constant pressure processing, and we do so by having no moving parts. So again, this interaction chamber is a fixed geometry. The microchannel does not change its dimensions throughout processing. And this varies a little bit from some alternative technologies, such as valve-based homogenizers, who focus on constant volume processing. And in order to do so, they have this small valve that is constantly opening and closing throughout the process to ensure this constant volume. So how this correlates when it comes to a pressure profile, as you can see in this, this graph on the top right, is with a microfluidizer processor, a vast majority of your material is being processed under the exact same pressure. So in this case, it would be a vast majority of your material would be processed at 30,000 psi. And while these valve-based homogenizers can likely reach similar maximum processing pressures, a vast majority of the material is actually being processed at all of these different processing pressures. So this correlates to a shear rate profile, as you might expect, where a vast majority of your material, when you are processing on a microfluidizer processor, is processed at the exact same shear rate, where again, with some alternative technologies such as this valve-based homogenizer, while they might be able to hit the same shear rate, a vast majority of your product is being processed at all of these different, I guess, ranges of shear rates. So how this correlates to your results is typically with a microfluidizer processor, you're able to achieve a smaller particle size with a tighter distribution in a fewer number of passes. So typically it is a much more efficient process. Additionally, as your material is all being processed under the exact same conditions, this helps to aid in batch-to-batch repeatability as well. So with that being said, I will turn the presentation back over to Vanessa.
Yeah, thank you. Sky Pharma, using this technology, has developed two bioavailability-enhancing solutions. So the first one is IDD Dissocube, which is able to decrease particle size down to micrometer to submicron range. And the final pharmaceutical form is microcrystals contained into either oral tablets or capsules. This technology is particularly suited for BCS class 2 molecule, or the one which exhibit a huge food effect. And this technology is already on the market and is already regulatory approved. The other one is more recent. It's called NanoMics, and it's currently being developed in partnership with La JEP, with Oksana being the PhD student. And it's further decrease the particle size down to nanometer range. The final pharmaceutical form is lipidic nanoparticles, either under oral tablet or capsules. And this technology is particularly suited for lipophilic BCS class 2 or 4 drugs. And as mentioned with Oksana, PhD student, so this technology is under development, preclinical development. So we will further explain these two technologies through two case studies. So the first case study will explain how IDD Dissocube technology may help Fenofibrate. So fenofibric acid is a lipid-reducing agent used to treat lipidic disorders. And the first oral dosage that was developed was a non-micronized 300 milligram capsule or tablets. The API used was a prodrug, fenofibrate, a poorly soluble ester, which was rapidly converted by tissue and plasma esterase into active fenofibric acid. It is also metabolized by cytochrome. And what was observed also that it had a huge food and fat effect that lead to unpredictable clinical efficacy. For instance, it was hypothesized that fat regimen were enhancing solubilization or also increasing hepatic blood flow, thus reducing first-pass metabolism. This led to a wide inter- and intra-individual variability. And that is why, for this specific drug formulation, non-micronized 300 milligram tablet or capsules, this needs to be administered with a fat food regimen to be really effective. So that is why TRIGLADE product was developed using IDD Dissocube technology to obtain small particles of phospholipid stabilized fenofibrate. And you can see on the image on the right, micro-sized cubes with an increased total surface area of the active molecule, thus enhancing bioavailability. So how is it manufactured? Through a 4-step industrialized manufacturing process with an overall batch size on 1,000 kilo and a process yield greater than 95%. So the first step consists in manufacturing a microemulsion of melted fenofibrate layered with phospholipids. So this step is performed at a high temperature to make sure that fenofibrate is maintained under fused status. And the phospholipid used is egg lecithin. So after 3 passes, the suspension is cooled down through a tubular heat exchanger in order to lead to the crystallization of fenofibrate. And also this new suspension is passed through a new microfluidizer at very high pressure this time to further decrease the particle size down to a micrometer range. So the resulting suspension is analyzed using Malvern for evaluating the particle size distribution, as well as an assay for fenofibric acid. So the resulting suspension is further mixed with the excipient to perform the drying using top spray granulation with a bulking agent being the lactose monohydrate. And after that, the resulting powder is compressed into tablets of 827 milligrams. So here you can see on this graph the clinical result of this new formulation using IDD Dissocube. What was observed into clinic is a similar bioavailability was observed with a reduced dose into the TRIGLADE formulation, as well as the elimination of the food effect, a faster onset of action, an improved absorption level, as well as an increased dose proportionality. So I will now hand over to Oksana for the second case study.
Thank you, Vanessa. So I am now going to present you our NanoMics research program, which is the topic of my PhD. In order to introduce this subject, Chris, could you please switch the slide on? Thank you. So I want first to introduce you the ideal features that we want for our nanoparticles, mainly based on the particle size analysis, which we can obtain with this kind of report, thanks to the nanosizer equipment. They concern first the mean diameter with our target value under 200 nanometers. Also, what we call the PDI, polydispersity index, which is the measurement of the particle size homogeneity in the mixture with our target value under 0.2. and also other ideal characteristic that we want. For example, spherical shape, because it is well described that a spherical shape is much more ideal for all the interactions on the lipidic nanoparticles, and of course for the liberation of the API. Good encapsulation efficiency, stability in digestive tract, of course, and other advantages due to this lipidic form. They offer controlled release of the API, which is really interesting in some cases. And they also offer a decrease in toxicity and food effect. So we are going to talk about SLN and NLC. So just to be sure to be clear, solid lipid nanoparticles, SLN, are the first generation of lipidic nanoparticles. And we have also NLC, nanostructured lipid carriers, which are the second generation ones. They have a really significant difference concerning first their lipidic matrix. About SLN, they only have solid lipid inside their structure. That's why they offer what we call the brick wall structure. And they have also an increased stability compared to other nanoparticles, for example, liposomes. What's different with NLC, first their size, they have quite smaller sizes, and they also have inside their matrix a liquid lipid, which offers an imperfect structure, not a brick wall at all. And thanks to this, they offer increased stability compared to SLN. When you want to entrap a drug inside your nanoparticles, against difference between those two types of lipidic nanoparticles, for SLN, there are low drug loading rates due to this brick wall structure because it leads to crystallizations issues. Also, during the process, it is possible to have a drug degradation. NLC, thanks to their imperfect structure, have higher drug loading rates than SLN, and also they are easy to scale up. And that is really an important point for us in this project, because we don't just want to lead research and development at lab scale. Of course, we're going to do this, but the main goal is also to scale up what we do at industrial scale at Sky Pharma. It is interesting also to notice difference during storage. Concerning SLN, a quite frequent instability phenomenon is what we call gelation tendency. In my opinion, gelation is not the perfect word because it's a bit confusing. I mean, we don't have any jellyfiant inside this formula, but really simply you have an increased viscosity of your preparation and you can't use it anymore after. Also, high drug expulsion, always due to this solid lipidic matrix. What's different with NLC is that they offer a better stability during the storage and also a decreased drug leakage. About NanoMics application, we lead what we call the nanosuit study, is to say selecting the right approach for your product. When we want to entrap a new API in our formula, the first thing that we do is we go and see if the API is stable at 70 degrees. Why 70 degrees? It's our work temperature, actually, because, as I said previously, we have solid lipid in the nanoparticles, but during the whole process, we want to have liquid phase. So in order to have liquid phase, we have to make sure to be above the melting point of the solid lipid. That's why 70 degrees, and of course, the API has to be stable at this temperature. After this, we prefer working with hydrophobic API, so with log P above 0, and also API which has issues with, for example, absorption and or poor intestinal permeability. or also high enzymatic metabolization. Really simply, API belonging to BCS classes 2 or 4. After this, we make sure, of course, that we can load in the NLC, in the lipidic nanoparticles, generally speaking, the therapeutic dose due and linked with the specific API. The last step is a solubility test in order to make sure that the API is going to be soluble in our current components of our formula. When all these steps are okay, we can go ahead on the formulation stage. Which is what is really interesting with the formula that we developed is that it is quite versatile, and so it allows a wide range of API encapsulation and also a wide range of solutions for patients. Concerning our formulation process, it is quite simple. We first do a simple emulsion with, on the one hand, the melted lipidic phase, so with all the lipids that we use and also the API. On the other hand, the aqueous phase with surfactant and water. After this, we do a first high-shear homogenization. When this first emulsion that we call premix is ready, we can start the Microfluidizer technology, so just as Chris explained it before. So we do the numbers of cycles that we want at a precise pressure that we chose before, of course. At the end of this process, we have a nano emulsion, a hot nano emulsion. But actually, we don't want a nano emulsion, we want a nano suspension. So in order to have this, we simply cool down the emulsion, and the solid lipid is going to solidify with the cool down. So during our first experiments, we pointed out two critical parameters due to the process. The first is pressure. As you can see here, we observe that increasing the pressure with the Microfluidizer technology leads to a significant decrease of the particle size parameter. Even for the mean diameter here in blue that you can see, and also for the polydispersity index in orange. In our case, we found out that 750 bars is really the ideal pressure to work for the preparation of the formula. Second critical point that we observed is the numbers of cycles that you do. Exactly the same experiments show us that increasing the cycle number leads to a significant decrease of the particle size parameters. In our case, 4 cycles is really the ideal numbers that we can do. Actually, as you can see, the results at 6 cycles are a bit lower, but actually there are no significant difference. And as we want to scale up this process, I mean at lab scale, it doesn't matter if you do 4 or 6 cycles, but at industrial scale, for economic reasons, there's a difference. That's why we chose 4 cycles to work for the formulation of our nano suspension. So to show you our first results, we first formulated blank nanoparticles, is to say nanoparticles without any API inside. So we did few formulas, first SLN1s, with particle size parameters really ideal. To remind you, we wanted the mean diameter under 200 nanometers and the PDI under 0.2. What is interesting is that thanks to transmission electron microscopy, we could see that our nanoparticles had spherical shapes. And as I mentioned previously, it is really one of our ideal features that we wanted. So all these little dots that you can see here on the slide are the lipidic nanoparticles at different scales. We did exactly the same with NLC formulas. So we did try several liquid lipids inside, for example, capriol 90 and Mazin CC. Quite satisfactory results too, even if the one with Mazin are better than the one with capriol. Thanks to microscopy, you can see also that our NLC has the expected spherical shape. After this, we try to encapsulate few API inside the nanoparticles. I am going to present you the results of the first one, which has diuretic properties with quite a high molecular weight, low log P, so not a very hydrophobic API, but still hydrophobic. Medium water solubility, in other words, it is a BCS class 2 API. So these are the results that we had for these encapsulated lipidic nanoparticles. As you can see here on the left, we obtained this kind of white nanosuspension at the end of the process. What is interesting to know is that even for blank and drug-loaded nanoparticles, we had satisfactory results concerning the particle size and also the spherical shape. It is really interesting to notice that API encapsulation improves the results, especially concerning the particle size. And we are currently working on the encapsulation efficiency because these results are quite satisfactory, but we are working on to optimize them, of course. We also studied the stability at storage of our formula. So as you can see here, during 7 days, the mean diameter of our preparation increased with time, but remains still acceptable. Concerning the polydispersity index, it is really interesting because we found out that it is much more stable and shows also a slight decrease. What we can learn with this stability at storage is that between the day we do and produce the nanosuspension and the day where we go on to the steps after, we can still have a stable nanosuspension. It's not an issue. The next steps are drying and tableting because, as Vanessa said previously, we don't want to do nanosuspension, but we want to formulate after this oral tablets. That's interesting because at La JEP we have a strong knowledge concerning drying of dispersed systems, and also Sky Pharma has a really strong experience in tableting. So I'm going to show you the first result of these two steps. Concerning drying, we are currently testing two techniques. So these are the powders obtained by the spray dry technique on the left and the granulation technique on the right. The general aspect of both of them were satisfactory. Residual moisture were under 4%. Both of them were easily dispersible. And they were also both easily scalable. So that's really a good point for the industrial scale-up. Concerning tableting, we were able to obtain nice tablets like these ones, easily dispersible too. And there were, of course, all the batches that we produced were, of course, compliant with the European pharmacopoeia controls. For example, disintegration time below 15 minutes, mass uniformity, hardness, and friability. So these are the results that we have now, but what's next? Mainly two studies. First one, an in vivo study, in order to show that our formula is able to enhance the bioavailability of the API. So an in vivo pharmacokinetic in vivo studies with mice by oral gavage. Second study is an in vitro one. in order to show that our formulation can enhance the cellular permeability of the API. Really simply, SLN and NLC can go through intestinal cells by two ways. First, the paracellular pathway, by opening the tight junctions, but actually this way allows only small molecules, few angstroms only. And there's another main pathway, which is the transcellular pathway with mainly endocytosis. And so for globally both of those studies, we want to compare the effect of our nano suspension, our nano powder, so the dry nano suspension, and compare these results to the free drug results too. And I'm now going to leave the speech to Chris.
Yep, thank you, Oksana. And as Oksana had alluded to, getting good results on the lab scale is a great first step when it comes to drug development. But if you want to be able to create a commercially viable product, really this entire drug manufacturing process has to be able to scale. And one of the major benefits of microfluidizer processor technology is that it is completely scalable. And we do so by placing the single microchannels that are housed inside of the interaction chambers on the lab scale in parallel inside of the interaction chambers on the production scale. By doing so and by maintaining the exact same geometry and dimensions for each individual microchannel that is in the interaction chamber on the lab scale on the production scale, all we are doing is allowing for a higher throughput through the interaction chamber while maintaining the exact same shear rate and impact forces that your material would experience on the lab scale on the production scale. And in doing so, as your material is being subjected to the exact same shear forces, the exact same impact forces, we can therefore guarantee that so long as you're processing under the same processing conditions, so that would include using the same interaction chamber, the same processing pressure, and processing for the same number of passes, we can guarantee that you will achieve the exact same particle size reduction on the lab scale and on the production scale. So speaking of scale, we offer a wide range of microfluidizer processors that are capable of handling a wide range of batch sizes. So from as small as 1 milliliter batch sizes with our smallest lab scale microfluidizer processor, up to flow rates of tens of liters per minute with our largest production scale microfluidizer processor. We also offer a variety of these machines in GMP compliant or CGMP compliant models. So really, regardless of your batch size, how frequently you might want to use the microfluidizer processor, or what regulatory requirements you might have, we should have a solution to fit your needs. I would also just want to point out on this slide that the two starred microfluidizer processors are the ones that are available via Sky Pharma and LabJep. So on the lab scale, they have an LM20 microfluidizer processor, which is really good for R&D batches. And then on the production scale, it's not this exact model, but it's a similar M700 series, which is our production series model. This model is GMP compliant as well, so it is capable of manufacturing industrial batches as well as clinical batches. Also, to briefly touch on some of the take-home messages as they relate to the microfluidizer processor technology. So our technology offers some unique features, including high potential processing pressures, and really the core of our technology, our fixed geometry interaction chamber, which work together to allow for constant pressure processing. And this constant pressure processing has some very good benefits, including that your material will be processed at constant specifically selected shear rates, which again can result in very small particle size potential, narrow particle size distributions, and as well as gives good batch-to-batch repeatability as well. Additionally, when we scale up, we take our single microchannel that is housed inside of the lab scale interaction chamber and create multi-slotted interaction chambers for the production scale systems. And in doing so, we are able to guarantee scalability.
So for the take-home messages for Sky Pharma and the technologies that are available here. So for the IDD Dissocubes, it's already market-approved technology. We have seen that a very high process yield. It is an industrialized solution able to handle batches from roughly 50 kilograms up to 1,000 kilograms. It's a process free of organic solvent, which is very important. And we can also, through this formulation, eliminate food effect of molecule.
And to finish concerning the NanoMics project, it is, in our opinion, really a promising technology for oral routes because it is really an innovative oral tablet formulation of SLN and NLC. What's more, it is really applicable to a wide range of molecules from several therapeutic fields. And there's also an expected protection against food effect and first-pass metabolism. That's why it will probably offer a lot of solutions for patients. Thank you for your attention. If you want further information, don't hesitate to write an email to us. You have our addresses here. Don't hesitate to follow us through our LinkedIn accounts. And I am now going to leave the speech to Aline, who is going to lead the question and answer session.
Thank you very much, Oksana, Christopher, and Vanessa for this presentation. So I am Aline Moulin, Pharmaceutical Development Director at Sky Pharma, and so I will be the moderator for this question and answer session. So we have some questions regarding first microfluidizer technology. So perhaps more for you, Christopher. How does this technology compared to ultrasonics to produce nanoemulsions and scalability?
Yeah, that's a really good question. So how ultrasonics works when they kind of reduce the droplet size of emulsions. So typically, these sonicators are probe-based sonicators that send out, very high energy sound waves. So how they, I guess just by their nature, when they send out these sound waves, a lot of the energy will be concentrated towards the probe itself, and kind of some of the material at the outskirts of your container might not be subjected to the exact same energy forces as some of the material kind of closer to the probe. So this can kind of lead to some issues in efficiency of creating nanoemulsions on the lab scale. And then kind of to answer your question on the scalability portion, essentially how these ultrasonic companies, I guess, scale up their product is by creating a bigger probe. And this is obviously not a directly scalable approach. So for example, you can't just I guess multiply the batch size and say this is 2x times the batch size. We might require 2x the amount of time to achieve the same results. Whereas with our technology, as we kind of discussed on a previous slide, by simply keeping the same dimensions of the microchannels and just simply placing them in parallel, we can ensure that you will achieve the exact same results on the production scale as you will on the lab scale. So really with our technology, scalability isn't an issue, whereas for kind of some of these ultrasound based technologies, you kind of have to re-optimize your process on the production scale.
Thank you, Christopher. Another question regarding the scalability of this technology. Would it be a 1:1 reduction in number of passes when moving from a single channel to 4 channels in production? So moving from 4 passes to 1 with 4 channels.
Yeah, that's a good question as well. And the answer is, so there isn't a reduction in the number of passes that you will experience. So essentially, your material isn't going to pass through all 4 of these microchannels. It's just the material will kind of separate when it enters the interaction chamber, so that I guess your material will pass through a single one of these microchannels. So when it comes to the number of passes, you will experience the exact same number of passes on the lab scale as on the production scale. So yeah, again, there won't be a reduction in number of passes, or typically it's in a reduction in number of passes. It should be the exact same number.
Thank you, Christopher. Some other questions relative to NanoMics platform. So perhaps Oksana, for you, perhaps just to explain a little bit more the difference of manufacturing process of NLC versus SLN that you presented slide 9. 19,
Yes.
I think. So that's perhaps to go a little bit more in the detail of the process.
Yes, of course. Actually, there's no big difference between those two type of lipidic nanoparticles. The only difference is that for NLC, nanostructured lipid carriers, we only add a liquid lipid. So for both of these formulas, we will still have the first emulsion that we did at the beginning of the process. The only difference is that when we are going to melt the lipidic phase, for the nanostructure lipid carriers, I am just going to add the lipidic phase inside this melted lipidic phase. So no much more difference concerning the process.
Okay. Another question concerning the tablet that you have manufactured. Have you tried to measure the particle size by dissolving sprayed dried API or tablet? So I think that is perhaps some result that you will have very soon.
Yes, I did actually. Well, I can't tell you exactly the results, but what I can say is that we could dissolve even the powder and the tablets in water after the drying and tableting process, and both of them didn't show any agglomeration issues. And we found that an interesting particle size analysis after, quite the same range that for the initial nano suspension.
Thank you. Another question related to the drying process that you have used to produce the powder. How is the nano suspension drying process achieved?
Really well. I just want to make sure to understand the question. You want to just to know if there are issues by drying the nano suspension?
No, I think that was just a question about what type of process are you using?
I'm using two type of processes. On the one hand, we tried spray dry. So with really a classical spray dry technique. And on the other hand, we did wet granulation with our nano suspension.
Okay.
On solid excipient.
I hope it answered to the question. Another question concerning the stability of the particles that you produced. Have you tried to check the particle size beyond 7 days?
For the moment, to be honest, no, but it's one of the topics of my thesis. But it will still be interesting to know, like after 1 month, 2 months, 6 months, how is going the nano suspension. But in our case and in our research program, we don't really need to know how is going the nano suspension after 1 month, for the simple reason that we are not going to wait 1 month between the nano suspension production and the drying after. And we know that when the nano suspension is dried, it is much more stable.
Thank you, Oksana. Another question regarding the process for the manufacturing of NLC. The cooling down of NLC after microfluidization, was it performed after collection of the material or during?
I do the cool down after I have the whole emulsion the whole nano emulsion that I wanted to. So at the end of all the cycles that I do, I collect this emulsion and I cool it down.
Okay, clear. And a last question concerning NanoMics platform. So Oksana, you showed a low impact of the number of cycles on the particle size and polydispersity. Why did you choose 4 as the optimal number of cycles compared to 2, for example?
For a few reasons, as you can see on the slide, thanks, Chris. At 2 cycles, we have a polydispersity index which is a bit higher compared what we want. And also at 2 cycles, it seems that it is, in our case, of course, it is not sufficient in order to have a stable formula. In our case, with our raw materials, the best numbers of cycles, which combines stability and good particle size parameter, is 4 cycles. That's why we did this choice.
And a question perhaps for Christopher, last question, what is the true part of average microfluidizer liter per hour? So of course it will depend on the model of the microfluidizer, but perhaps Christopher, you could elaborate a little bit more.
Yeah, and you're exactly correct. It depends on the model. So obviously we have lab scale models that have, slightly lower throughputs than some of our production scale models. I will say on the production scale model, if you do need the full 30,000 psi of processing pressure, the maximum throughput you might be able to expect on one of our production scale machines would be around 5 liters per minute. But what we do with our production scale units is we can actually cap them at different maximum processing pressures. So we have a 10,000 psi unit, a 20,000 psi unit, and a 30,000 psi unit. And if we are able to cap the maximum processing pressure at a lower pressure, so 10,000 psi, for example, we are actually able to get a higher throughput. So it really depends on the actual model you choose and the processing pressure you select. But I guess as a maximum, you might be able to expect somewhere in the range of 10 to 15 liters per minute. And then obviously we again have models that are smaller pieces of equipment that can do lower throughputs as well.
Thank you, Christopher. Is there some other questions? So if no, we will thank again Oksana, Christopher, and Vanessa for this presentation, and thank all of you for attending this webinar. Thank you very much, and we will you will receive in a few hours